Bismillahirrahmanirrahim. Yesterday we discussed in detail the overall process of translation that was divided into 3 steps in initiation, elongation and termination. This step is of prokaryotic process of translation, the synthesis of protein synthesis in case of prokaryotes. So if we want to stop this prokaryotic synthesis, we need to know the action of different antibiotics in these steps of translation, where the overall process of translation is going to be inhibited with the help of some diseases, some medicines. So streptomycin is the first drug, and the mechanism of action of streptomycin is it binds to the 30S subunit and distorts its structure, interfering with the initiation of protein synthesis. So what is the mode of action of streptomycin? It is going to bind with the 30S subunits of ribosomal subunit. If its structural similarity is like 30S subunit, then it is going to inhibit the initiation of translation. So overall growth of bacteria should be inhibited. Then comes the second antibiotic drug, that is tetracycline. It interacts with the 30S ribosomal subunit, blocking access to the amino transfer RNA to the A site of the messenger RNA ribosome complex. So ultimately tetracycline also binds with the 30S subunit and specifically at the A site of the 30S ribosomal subunit by inhibiting the amino acid transfer RNA synthesis. So no upcoming amino acid transfer RNA will be coming and attach at the A site, and no amino acid is going to be added to the growing polypeptide chain. So no protein synthesis could be accomplished. Then comes the action of tetracyclines. Tetracyclines interacts with the 30S ribosomal subunit, blocking access of A site. So we have done this tetracyclines, and now we are moving towards the puromycin. Puromycin bears the structural resemblance to the amino acid transfer RNA and becomes incorporated into the growing peptide chain. So the mechanism of action of puromycin is it has structural resemblance with the amino acid transfer RNA. So it binds with the A site in place of amino acid transfer RNA and ultimately blocks the polypeptide synthesis chain. Then comes the chloramphenicol. It inhibits prokaryotic peptidyle transferase. High levels may also inhibit mitochondrial protein synthesis. So when there comes a point when the amino acids are going to be joined together with the help of peptidyl transferase and peptide bond is going to be formed between them, then chloramphenicol is going to inhibit the action of peptidyl transferase, thereby inhibiting the synthesis of prokaryotic protein synthesis. The last two antibiotic drugs are clindamycin and erythromycin and diphtheria toxins. Clindamycin and erythromycin binds irreversibly to a site on the 50S subunit of the bacterial ribosome. So we studied streptomycin and tetracycline to be the structural similarity with the 30S subunit, while clindamycin and erythromycin having structural similarity with the 50S subunits, and it intercalates with the translocation process. So the last drug is the diphtheria toxin. It inactivates the eukaryotic elongation factor EF2. So no energy will be transferred to complete the translocation process, and thereby inactivation of eukaryotic elongation factor leads to the inhibition of protein synthesis. After the process of protein synthesis completed, we must know these different types of medicines which are there, so we can stop the synthesis of these microorganisms, especially bacteria, in our body. After that, we observe many co- and post-translational modifications which are basically important to define the destination of these proteins. So co-translation modifications are those modifications which are done on the proteins when still attached with the ribosome, when the machinery is not distorted and the polypeptide chain that is newly formed is attached with the ribosome. And these co-translational modifications gives a clue or gives the destination to that protein to move that particular point. And after the polypeptide chain becomes detached from the ribosomes and transfer RNA, the post-translation modification occurs on them. These post-translation modification could be trimming or covalent modifications. Trimming is just the cutting of different areas of the protein so that it may fold into different shapes and go to their destination, while covalent modifications involves the attachment of different chemical substances or chemical structures to that particular protein to define its destination. These covalent modifications could be phosphorylation, glycosylation, hydroxylation, and other biotinylated or furancylated. So in case of phosphorylation, you must know that phosphorylation is the attachment of phosphate group, and if the phosphate group is going to be attached with the protein, that protein is going to be activated or inactivated according to the nature of that particular shape of the receptor on which this protein is going to be attached. So phosphorylation is the attachment of phosphate group, and this phosphate group attachment define the destination of that particular amino acid or protein. Then coming towards the glycosylation part, if a glycosylated groups are going to be attached with different proteins, then their destination must be different. Like in case of N-acetyl glucosamine and N-acetyl glucosamine are going to be attached with the proteins, then they must be some type of structural similarity with the cell membrane proteins and many others. Then moving towards the hydroxylation and carboxylation attachment. This hydroxylation and carboxylation groups attachment basically involves the hydroxyl groups in case of hydroxylation. And remember, in the vitamin chapter, in the vitamin C, hydroxyl groups are attached with the prolyl residues, making them hydroxyprolyl residues. So these hydroxyprolyl residues are now becoming functional for the activity or active vitamin function. And in case of carboxylation, remember in vitamin D that carboxyglutamyl residues are being carboxylated with the help of attachment of carboxyl groups to make them to convert the immature clotting factors into the mature one, including clotting factors 2, 7, 9 and 10. Then different biotinylated groups and furancylated groups are attached in different vitamins and lipids also. So the basic purpose of this co and post transition modification, that we are making these proteins able to go to their destinations, as you know that different proteins are going to function in different tissues differently. if a protein is there in one tissue, then there must be an isozyme of that protein in the different tissue of the different organ. So these co and post translational modifications are very important, and these are the clue for the proteins to go to their proper destinations. And finally, these PTMs are very important according to examination point of view, because examiner do not always ask all the steps of translation. It may ask you like the question of antibiotic resistance, which antibiotic is going to inhibit which part of the translation machinery, and how many types of covalent modifications are there in PTMs or in post-translation modifications. These questions make sense of about 1 to 1.5 marks. So keep in mind, these short questions may come in your exam, and these are very important according to examination point.